Thanks very much for that introduction. I'll discuss today, as mentioned, the needs and opportunities of radioligand development in neurologic and psychiatric research. It's a daunting task, and I don't think I have a complete overview, but what I can do is give you my personal opinions based upon what I have done and what I am planning to do, at least in my individual career. In that regard, let's advance the slide. This is a diagram of what my career has sort of been like up till now. We developed many PET radioligands. In this case, it was a radioligand called PBR28. That is, it's a biomarker for TSPO, translocated protein, biomarker for inflammation in the brain. It's highly expressed in activated microglia and reactive astrocytes. When we first developed it, we looked in a rat stroke and we found that we could see the known inflammation surrounding a stroke in the middle part of the striatum. We moved on to monkey studies and we could do full quantitation in the brain, and this number that we derive, VT, is linearly proportional to the density of target sites. We go on to healthy human subjects, and this image reflects the density in each voxel, showing that we can do full quantitation using arterial blood samples and brain imaging. And along the way, we had a, quote, healthy subject referred from Hopkins, who in fact had this bright red spot, and we thought it might be, and it was, a small stroke, lacunar stroke, that the subject had had about 2 weeks prior to the PET scan. So the scope of my work has clearly been translational as it goes from a rat stroke to a human stroke. But where do I want it to go, and what would, based on that, my recommendation to the field be? And it's trying to link these numerous PET radioligands that we've developed to treatment trials to better understand pathophysiology, but most importantly, to improve treatment. So for example, with this biomarker of inflammation, and as a psychiatrist, I could ask, do a subset of patients with depression have neuroinflammation shown by PET imaging, elevated TSPO? And if that's true, can an anti-inflammatory drug treat depression in the subset with neuroinflammation? This is really asking the question, can PET be used as a biomarker to predict treatment response? This is precision medicine or patient stratification or personalized medicine. And the answer to this question is, I can tell you quickly, is that do a subset of patients have neuroinflammation? Yes, 6 out of 7 studies have reported elevated TSPO in unmedicated depressed patients during a depressive episode. Can an anti-inflammatory drug treat depression in this subset with neuroinflammation? And there was a report just last year by Jeffrey Meyer. He did TSPO imaging at baseline. He treated the subjects for patients for 2 months with a COX-2 inhibitor, celecoxib, an anti-inflammatory drug. And there was a modest response. But it only occurred, it was almost exclusively occurred in those individuals who had elevated TSPO at baseline. So it may be that PET can be used as a biomarker to predict treatment. And that's where I want my work to go. So let me just explain first what TSPO is, and then I'll summarize the studies in depression. TSPO is a biomarker for neuroinflammation. It's a translocated protein, 18 kilodalton. It's located in the outer mitochondrial membrane. Its functions are unclear, but may be involved in steroid synthesis. And lesions increase TSPO over a few days, and that elevation may stay for weeks or months. So if you did an electrolytic lesion or an inflammatory lesion, inject LPS into the brain, it takes about 3 days to reach a peak level, and then it can stay for weeks or months. So it's relatively slowly responding and relatively long lasting. Its cellular localization of TSPO is in microglia, astroglia, as well as vascular endothelium. Okay, so the studies in major depressive episode in patients during an episode of depression 6 of 7 studies in unmedicated MDE showed a widespread increase of TSPO. This is reviewed in a recent review article in Lancet Psychiatry, and there's a companion article in Lancet Neurology, looking at PET imaging of neuroinflammation in psychiatric and neurological disorders. So 6 out of 7, that's very strong. That's something you can hang your hat on, I would say. However, there was no correlation with the severity of illness, but the increase may be related to the duration of untreated illness. So it may be related to the burden of total inflammatory time. TSPO is long-lasting. It could be a cumulative measure. Is it a predictor of treatment response? Well, very importantly, this study that I mentioned from Jeff Meyer, Atwells was the first author. Celecoxib had a modest treatment effect, but it was largely restricted to patients with elevated TSPO at baseline. Okay, so that is, I think, a really important study. I hope that it is for more to come. Celecoxib, I might mention, is an inhibitor of COX. It's a preferential inhibitor of COX-2, cyclooxygenase 2. Okay, so this has been done in major depressive episode. Let's say, okay, shall we do this in patients with Alzheimer's disease? Because Alzheimer's disease is known to have elevated TSPO. So the question I want to address, the second half of my talk, should we do such a study in patients with Alzheimer's disease? Well, here's a summary of the TSPO imaging studies in Alzheimer's disease. This comes from a review article that was published in 2019. I've done individual studies, but I think if you want like the true story, you go to some review article. And it's very clear that it is a strong marker of disease presence. If you have the Alzheimer's disease, you're going to have elevated TSPO, and it's known at the time of death that there's neuroinflammation. That's a 3+. Does it correlate with disease severity? You know, cross-sectionally, you know, across individuals, if you have more severe cognitive impairment, do you have more? I would say that's about a 2+ correlation. And then does it correlate with disease progression, a biomarker of disease progression longitudinally? I think there's only one study, and that was our study, so I'll give it just a single plus. So the patients had an increase of TSPO over 2 to 4 years. Healthy subjects did not. And the increase in TSPO correlated with the increase in cognitive impairment. So this is promising, but just a single report, so I'll give it a single plus. So could TSPO imaging be used to predict treatment to an anti-inflammatory? That has not been done, and there's only one report in which that was done in the case of depression. And when I've thought about it and also discussed with pharma people, it's unlikely this is not what's needed now. So in this thought process that I'm going through with you, it's like, well, what biomarker is needed? And the reason why we're not ready to do this study, or at least, well, I wouldn't do it, the field may not be ready to do it, is that the treatment improvement in depression can occur in 2 months, but the cognitive improvement or the slowing of cognitive decline may take years. So if you had some novel anti-inflammatory drug and you wanted to give it for a couple of years, you wouldn't want to do that to predict the final treatment. Instead, before that, you would want some biomarker that would show that you are having an anti-inflammatory effect on the brain. So I think the biomarker that's needed in Alzheimer's disease is not one to predict treatment, because the treatment period is going to have to be too long, but maybe one that would reflect treatment. It's not, and that has not been done. And I don't think it'll work with TSPO, because TSPO stays for a very long period of time. It's long lasting and relatively cumulative. So in that regard, what I would recommend as a biomarker to reflect treatment response would be COX-2, cyclooxygenase 2. And we developed radioligand selective for the 2 isoforms, COX-1 and COX-2. The radioligands are potent and selective for the appropriate target, as shown here. And as some further background, as a general rule, COX-1 is considered constitutive and it's present in microglia in the brain. Constitutive means that it's present at healthy tissue. In contrast, COX-2 is inducible, present at relatively quite low concentrations in healthy tissue, but it's markedly inducible. It can go up within a couple of hours in the periphery and come down in a couple of hours if you remove that. And its localization in the brain includes in neurons and microglia. But it is a fast-acting response, and it does show response to anti-inflammatory treatment. So here's the imaging of the COX-1 radioligand. COX-1 is constitutive, it's present in many organs of the body, including the spleen, the GI tract. And then here we have a block study. This is with ketoprofen. Ketoprofen is a very potent and selective inhibitor of COX-1. In the brain, COX-1 is widely distributed throughout the brain, but there is also an increased concentration in the hippocampus, the occipital cortex, and the pericentral cortex. Because COX-2 was a far more difficult radioligand to develop because there's very little COX-2 present in, let's say, healthy monkey brain. So in order to see whether our radioligand worked in monkeys, we had to induce an inflammation. And actually in a monkey brain following a lesion, there was a hemorrhage, and bleeding causes a marked inflammatory upregulation. And when we had that, we could measure in the monkey brain, not in the healthy monkey brain, but in the model of an inflammation in a localized area. And it could be, blocked by COX-2 selective compounds. So that looked promising, but does this COX-2 radioligand work in human subjects? Again, in healthy subjects, it's very hard. In healthy monkeys, it's very hard to see anything, except in the ovary, I should mention. So to see whether the radioligand worked in any human condition, we looked at rheumatoid arthritis, and we found elevations in the joints, the symptomatic joints, the currently symptomatic joints in patients, and it could be blocked, partially blocked. We have to go to a higher dose of celecoxib to block it more. And I think a very informative point with this was that we also did TSPO imaging. TSPO imaging has been previously found to be elevated in rheumatoid arthritis. It's present in macrophages, for example, and leukocytes. And it stays for a long period of time. But the COX-2 was elevated only in the symptomatic joints and not in the asymptomatic joints. TSPO was elevated in both. So this shows how COX-2 can be a marker of current inflammation, which presumably will go down if it was treated properly. And that's what we're going to look at the patient's rheumatoid arthritis before and after treatment. However, we expect that it would come down because look at these other asymptomatic joints. Okay, so in discussing this with some pharma partners, it seemed that what would be needed now, and this is sort of like the thought process I'm going through, would be some biomarker to show, to reflect treatment. And COX-2 seems like a possibility based upon that is the case certainly in the periphery, in joints. So stepping back a bit from trying to use it as a biomarker to predict treatment response, a proposal that is under discussion with Takeda, Janssen, and NIMH would be to look at 3 targets, COX-1, COX-2, and another one that I have not mentioned, CSF1R, colony stimulating factor 1 receptor, which is exclusively located on microglia, and a radioligand has been reported from Hopkins on that. We would look at those 3 targets in 3 disorders, Alzheimer's disease, multiple sclerosis, and depression. This would be a multi-site study. And the question is sort of like, which biomarker best reflects neuroinflammation in each disorder? We're going to have to define what best means. Stepping back a bit then, what I'd like my research to do, and what I think would be helpful in the field, is that the numerous radioligands that are developed for the brain be used to really critically inform pathophysiology and, in the best of all worlds, lead to improved treatments. I gave an example of a target that we've studied, TSPO, which seems promising as a biomarker to reflect treatment response in patients with depression to a novel treatment, which would be an anti-inflammatory. In my opinion, and that some others, we're not ready to try that in Alzheimer's disease. Instead, because it's going to have to be a long trial, what would be helpful is some sort of a biomarker to reflect the treatment. So we know that the treatment was working and active in the brain. And one possibility for that would be COX-2, perhaps also CSF1R. And I don't know about COX-1, it just hasn't been, I haven't seen it. I certainly in the brain, I don't know. Okay, so this is how I've proceeded that. I would proceed with that. Thanks very much for your time.